Hi friends, we are going to discuss about the DNA repair mechanisms. DNA repair mechanism. DNA repair. What is first? We have to know what is DNA repair. It's a collection of process by which a cell identify and corrects a damage to DNA molecule that encodes genome. Means that DNA damage occurs maybe many reasons due to mispairing or some mutations for any abnormalities. The DNA will be damaged due to some of the high temperature condition, denaturation like that. DNA can damage many ways, but this process to repair means we are going to repair what is the identify which where the DNA is damaged, and we are going to correct the damage is the DNA molecules. This is called DNA repair. And in this video we are going to discuss exercise repair pathways for the DNA repair mechanisms, and in further videos I am going to do on the repair of double stranded breaks or direct reversals. Base excision repairs. In this base excision repair mechanism, first one of the strand is deaminated C, where we can find here deaminated C, and with the help of there is a wrong base pair between the in the DNA segments. With the help of some of the enzymes like DNA glycolysase and AP endonuclease and deoxyribose phosphodiesterase enzymes, with the help of DNA glycolase enzyme, it's in this segment there should be a C instead of here. So we have got U in the DNA segment. So C, that is cytosine is deaminated. So that's why with the help of the DNA glycolase enzyme, which detect the AP site, which is going to detect the pyrimidine site in the DNA segment, and after that it is going to detecting the AP site at a pyrimidine site. That AP endonuclease just cuts the DNA segments where that DNA glycolase enzyme detected the AP site. That the cutting or cleaving of the segment which is mutated, that is deaminated, it's done by AP endonucleases. And after that, with the help its AP endonuclease enzyme sites with a missing base cleaves the sugar phosphate backbone. The sugar phosphate backbone present in the DNA structure, it's going to break down by the AP endonuclease, and next, and this deoxyribose phosphodiester removes the sugar phosphate lacking base. And as I said, AP endonuclease breakdowns or cleaves the sugar phosphate backbone, whereas deoxyribose phosphodiesterase removes the sugar phosphate lacking the base. And with the help of DNA polymerase enzymes, it's a very important enzyme which is helps for pairing of complementary base pairs, that joins the cytosine for the where the mutated or deaminated region is there. In that place, the cytosine added by the polymerase enzyme, and the other one is a DNA ligase enzyme, which joins the whatever the cleaved segments or the parts of the DNA, which is joined by the DNA ligase enzyme. Nucleotide excision repair. In this method, the pyrimidine dimers which are formed by the UV light, that is DNA damage is caused by UV light, that is formation of pyrimidine dimers. In the nucleotide excision repair mechanism, which is mediated by UVR proteins, which are most important for the repair mechanism. In this we can find that pyrimidine dimers which is formed here, TT, which is called a pyrimidine dimers, and the UVA proteins and UVB proteins that walks on the segment of the DNA and find out where the pyrimidine dimers are occurred, and in that place which is UVA protein, UVB protein, this complex is going to find out where they track along the DNA in search of damaged DNA, where the DNA is damaged. After detecting pyrimidine dimers, the UVA proteins are released and UVC proteins binds to the UVB proteins, and And in this protein, UVC protein, which is makes cuts on the both sides of the thymine dimer, which is, as I say, pyrimidine is thymine dimer, which cuts the surrounding portion of the dimers. Usually the UVC proteins, which is cuts the 4 to 5 nucleotides from the 3' end of the damage and 8 nucleotides from the 5' end of the DNA. And after the cleaving that segmented double, what do you call that, pyrimidine dimers, which is having a pyrimidine dimer where the DNA strand is having, that is removed with the help of cleaved with the help of UVC proteins. And after we are getting with the help of DNA polymerase enzyme that fills the gap, and the DNA fills the gap that formation of complementary strands to the segmented segment, whichever the here in this region, it's going to add the complementary nucleotides with the help of DNA polymerase enzyme. And the DNA ligands which seals the gap, like breakage, it's going to seal the gap of the segment of the DNA. And by this we can see that there is no pyrimidine dimer, that is thymine dimer, occurred in the segment of the DNA. Thus the repair mechanism is completed. Thank you. Please subscribe our channel. If you want that other two types of DNA repair mechanism, please see our other videos. Thank you.